Hi, everyone. Emily Grason, the woman, the woman who can title her talks something like eunuch zombie snails, because she studies those, and who hosts the blog site Rah Rah Radula. And we have been blessed with her presence for the last, years. All right, I can go forward with this. I can, but I bet I have to press this one. Nope. That's what I tried.
Oh, hang on, you guys. Let's try this. Naturally.
And she has every piece of technology going up here so that people worldwide can actually listen to her. All right. So this morning, I did just a little surfing on the web to find the electronic virtual presence of Emily, which is pervasive. But I also realized that almost all the pictures of her are of her in the mud on her hands and knees, looking at something very closely. So she writes for SciPause. She put together the website for the Green Crab Citizen Science Monitoring Program. She has been a leader in writing for the graduate students on biodiversity science through Biodiverse Perspectives, which has enabled her to speak with really the leading lights in ecology and to interact one-on-one with them. And she also translates science speak into public speak through her internship with a funding agency, the JRS Biodiversity Foundation. So she has been extremely busy in many ways, in addition to her thesis. And I don't even think she's going to tell us all about her thesis today. I will simply say that I think she will emphasize the chapter that essentially takes 7 thesis chapters and puts them into one. Like most people would go find those 7 projects done by 7 different people and do it as a meta-analysis, but Emily decided she had to do all 7 of those projects herself and then look for generality, which amazingly she found. Spoiler alert, yes, all right. So with that, Emily, please tell us about your PhD.
Thank you. Thank you guys all so much for coming. Now I will see if this actually does work. Slide advancer. Might not. I might have to do this manually. That works though. Well, in any case, so I wanted to make sure that I thanked you for coming today because all of you have taken time out of your schedules to come and hear me talk. So I wanted to make sure you walked out of here with at least one interesting piece of information that you could tell people over your Memorial Day weekend barbecues. So first, I share with you this undeniable truth that I found on the internet, which is that Washington State has more species of invasive snail than any other state in the country. I know it's true because I found it on the internet. So this is something to be proud of. Say what you will about whether it's a surprise that Iowa produces more hog waste than any other state, but I think it's interesting that Washington State has a lot of invasive snails. Now, undoubtedly, this tally includes both freshwater and terrestrial gastropods as well, but a large part of our achievement in invasive gastropods, I think, probably has to do with our rich history in aquaculture. Charles Elton was one of the first to recognize that oyster culture in particular has been a hugely important vector in moving species around the planet. And the Pacific Northwest has been a really good case study. So our single species of native oyster, the Olympia oyster, the fishery collapsed in the mid-1800s due to a combination of overharvest and pollution. So oyster growers started importing other species from around the world to see if they could get anything else to grow here. And one of the prime candidates is this guy. This is the Pacific oyster, which, notwithstanding its common name, is actually not native to this area. It's native to the Western Pacific. And this is the species you're most likely to be eating 9 times out of 10 if you're eating a given oyster. It's by far the most widely cultured worldwide and certainly comprises the bulk of the commercial oyster industry in Washington state. So we import these oysters, and we accidentally import a lot of other species, including this contribution to the gastropod diversity of Washington state. So these 6 species are here as a result of oyster culture in Washington state. These are my mollusca non grata. And if you snuck a peek at their common names, you see they come from 2 major regions of the world. We have the eastern dogwhelk and the Atlantic oyster drill from the east coast of the United States. They were imported with the eastern oyster, Crassostrea virginica. And the rest of the species over here came from the western Pacific on imports of the Pacific oyster, Crassostrea gigas, or gigas if you're a Latin pedant. So this history tells us something about how all of these species ended up getting here, but I'm interested in the question of how they end up being able to stay here. What are the factors that enable them to survive in this new habitat? Because if you think about it from the perspective of a non-native snail, it's kind of a daunting proposition to show up in a new place where you have no evolutionary history and survive. So to suggest an analogy, being dropped off the boat in a mudflat in, say, Samish Bay is a little bit analogous to traveling to a new place without having done any preparation for your travel whatsoever. So these undoubtedly well-meaning travelers, I'm sure, have several questions, namely, what is this? Is this food, and how do I eat it? What is up with the local fauna? And how do I communicate with the other locals in the area? Because often the paradigm of evolution is explained by evolution as the process of natural selection, resulting in the high adaptation of species to their environments and to other organisms in the environment. So how is it that these guys make it? How do they survive when they have a relatively limited evolutionary history? They show up, they have no friends in the area, no couch to crash on, and they not only survive, but in a lot of cases, they manage to thrive. So this is a photo that comes courtesy of George Holm in the Pacific Northwest Shell Club, who I think is actually listening out there. This is Batellaria atramentaria, which can achieve these astounding densities. This is an impressive picture, but it's not actually that surprising or uncommon. So in particular, I'm interested about how these species of snail cope with our unfamiliar predators. We are world famous here, if I do say so, for our crabs. And in particular, we have a diversity of crustaceans that should be able to control populations and distribution of these invasive snails. I put Dungeness on top because they're everybody's favorite, but they're also pretty big and relatively abundant in the subtitle. And red rock crabs are another species that are big motivated predators on gastropods. So these should be species that should take care of these snails. How do we get situations where there is such astounding abundance? So the approach I've been taking is to ask what the role of evolutionary novelty is in this issue. So the concept of evolutionary novelty is pretty important in biological invasions because, as I alluded to before, an organism arrives and is surrounded by an environment and other organisms that it may not have evolved to cope with or recognize, etc. Biological invasions then give us the opportunity to study the role of evolution in species interactions by taking some of the evolution out of the equation to see what happens. So, my dissertation research has tried to delve into this question by asking how are these non-native snails so good at not getting eaten? Right now, I'm going to go through and outline each of the 3 chapters, but as Jen mentioned, I'm just going to talk mostly about the 3rd chapter. So chapter 1 focused on the question of novelty as a double-edged sword. So a non-native species gets here, and it's surrounded by unfamiliar organisms. But similarly, it is itself an unfamiliar organism to all of the resident species. So in a field experiment, myself and my co-author, Eric Buell, who's hiding behind Ellie, we assessed the role of non-native Japanese oyster drills, aka Godrilla, on oyster beds in Washington state, both as a novel predator for native oysters and as a novel potential prey for native crabs. In chapter 2, I looked at this question of the really astounding abundances that can be attained by the Asian mud snail batellaria, and comparing sites where batellaria are hyperabundant to where they're less abundant. If we look at different factors such as novel enemies, familiar enemies, and residence time, can we disentangle what factors enable them to be more successful across different contexts? And the third chapter, what I'm going to spend most of today talking about, is about how non-native snails assess risk from a novel predation threat. So, this project, I would call it like a study, but as Jen mentioned, it was like an agglomeration of a bunch of studies, so I've taken to calling it a project. This project, what it did is it really zoomed in on predator-prey interactions between crabs and snails, asking how the prey perceive the crabs. How do snails tell they're in danger from an unfamiliar predator? And how do they make the decision about how to defend themselves? Going back to the idea about novelty in species interactions, I might have the expectation that non-native prey would have a difficult time recognizing risk from a novel native predator. And we've seen good evidence for this in a number of invasions. The best known, of course, is the brown tree snake on Guam, which extirpated many local and endemic prey because those prey had not evolved to recognize and respond to a nocturnal arboreal tree snake. And as you know, if you have met or were at Evans Defense, that has ripple effects in the community where the loss of seed dispersing birds can change the plant community, and the loss of insectivores does good things for the spider population. There are a lot of other good examples. The Argentinian fire ant is invasive across a lot of the United States, and this species will attack birds' nests and consume the young. And in case you're ever in this situation, if your nest is attacked by an Argentinian fire ant, you should flee and desert your children and go on to renest elsewhere. But there are native birds that fail to do this. They stay and they try to be heroes and defend their babies, but in the process, they get injured and reduce their fitness for renesting. Another example I want to cover is a native crayfish in New Zealand, which does not respond defensively when it smells chemicals coming from invasive brown trout, leading to the possibility of increased mortality from predation. So this was the story for a long time in biological invasions, where there were scary invasive predators coming in, and the prey had no clue what was going on, so their populations really got hammered. But there are more examples of when prey are able to recognize novel predators. For instance, on the east coast of North America, the invasive European green crab sheds chemicals, and the native mussels need only to smell the chemicals from the crab to know that they should thicken their shells to protect against the crab's crushing claws. And closer to home in Oregon, invasive bullfrog tadpoles change their behavior and hide when they smell native predatory fish, which increases their survival. And there's invasive Daphnia in lakes across the country, our country, North America. And this species in particular grows big head spikes and tail spikes when it smells native predatory fish, and that reduces the ability of fish to prey on it, which increases its success as an invader. So there are cases here both where recognition succeeds and when it fails. What's going on here in terms of why we sometimes see success and why sometimes we don't? Well, I think first, one of the key things to consider in understanding why non-natives sometimes fail to recognize predators is to consider what happens in a co-evolved risk assessment. In general, how do prey tell that they are in danger of getting eaten? So at the heart of any risk assessment is a fitness trade-off. Prey can't run and hide all of the time. They can't live with their heads in the sand because they might miss out on lunch. That is, these defenses might increase the probability of avoiding or surviving an encounter with a predator, or they should increase the probability. But they also come at the cost of engaging in other fitness maximizing activities like feeding and reproduction. So optimally, prey should wait to defend themselves until they have enough information indicating certainty of risk to offset the cost of defending themselves. They want to wait to be sure. So how do they actually make these risk assessments? What information do they use? Well, there are a couple of common types of information that prey receive, and common modalities are auditory, visual, and chemical signals. Sometimes mechanical or tactile signals are also important, especially for plants. But these signals can come from several sources. They can come from either the predator or from other prey. So this prairie dog might hear a coyote or might hear an alarm call from another prairie dog. Similarly, that prairie dog might see or smell the coyote, or see or smell another dead prairie dog, indicating a nearby threat. So how does all this information get put together to make a risk assessment? Well, these cues aren't all equally valuable to risk assessments. Remember that in order to defend, a prey wants to be certain that it's actually about to get eaten. And one of the important qualities of the signal is the amount of information that it contains. So if there is more information in a signal, then prey can have a more precise picture about the nature of the threat. So here I'm going to walk through an example hypothesis of how we might think some of those signals from the previous slide relate to each other in terms of information. And I'm going to move into a crab-snail system here, since that's what I do. And in crab-snail systems, chemical cues are basically the most important. By the time a snail is either touched by a crab or sees a crab, it's not going to be able to outrun or defend itself. It's just too slow. So I'm talking about chemical cues between crabs and snails here. Now, if you are a snail oozing along the bottom, and you suddenly smell another snail of the same species, and that's called a conspecific. I'll be using that word quite a bit for the rest of the talk. That might pique your interest if you smell one of your conspecific friends that's been crushed or damaged in some way, but it doesn't provide you with a lot of information about whether you are actually in danger or how to respond. Now, chemicals released by injured snails are often called alarm cues, and they're considered to be general information because the receiving snail basically only knows that there was some kind of threat generally nearby, but not much more than that. Now, if that snail smells a crab, that gives it a lot more information. Namely, it knows where the predator is, what kind of attack it might expect, and sometimes they can tell something about the motivation state of the predator. Cues that are shed by the predator and are informative to prey in risk assessment are called chiromones. Now, you can get more information if you combine cues from crabs and from other injured snails. So if a snail smells crabs eating other snails, it not only it gets access to both the alarm cues and the chiromones, and it knows that maybe that crab is now interested in eating snails like itself and capable of doing so. So in terms of the value of the different information sources here, cues that offer more specificity should be more valuable to risk assessment because they increase certainty. On the other hand, it's often argued that responding to general cues like alarm cues could be disadvantageous because they don't tell you what to do. So if you don't know how to respond, you could actually respond in the wrong direction and increase the danger that you're in. Or you might over-respond and miss out on lunch. So more information should increase the probability that the snail chooses the right thing to do, and that should increase their investment in the defense. However, to complicate the picture, there are actually several pieces of chemical information that change simultaneously in this big gap that I've left here, and those result from the interactions between the snail and the crab. So what you have here is digestive enzymes that are being secreted by the crab that could also be influential in decision making. And you also have post-digestion snails, which is to say snail-flavored crab poop. And both of these things are being added to the interaction at the same time. So to disentangle the contribution of those two components, I'm going to consider two additional scenarios. First, that the chemicals of the crab are present at the same time as the chemicals from the injured snails, but they're not interacting with each other. So you can just add those two external cues together. And you might think that you could figure out how much time or how much defense prey should invest in response to the combination of cues just by calculating the responses to the cues when applied separately. But it turns out that people almost never test that. So it's a worthy sort of scenario to consider here. A second scenario is that the crab is eating something, but it's sufficiently unfamiliar or unstinky to the receiving snails, so it doesn't have any alarm cues or post-digestion snails. In my experiments, I was feeding frozen fish fillet to the crabs. Would someone mind actually closing these doors? I don't think we're going to get any more folks. Thank you. So to me, it's not really clear that these two digestion treatments, one is more informative than the other. I have to say that I somewhat lump them together in terms of my hypothetical information spectrum, but for the sake of having to exist in 2 dimensions here, they are plotted as such. Now, all of these scenarios have more information than if there were no chemical cues in the water. And so certainly for the sake of completeness in any experiment, I'm going to have to include a control treatment that doesn't have any chemical information or any added chemical information. And that'll help me establish a behavioral baseline. So this is an expanded, higher resolution way to test the theory about that more information should translate to higher value defenses and more investment in defenses. It's also a novel framework in which to test the idea of evolutionary novelty. So if you don't have access to the signals that are coming from crabs, you could potentially lose a lot of resolution in your risk assessment. So researchers have actually suggested that non-native snails have a workaround for this. That essentially, if they are overly or generally more sensitive to these cues, alarm cues from crush conspecifics, they would actually be able to protect themselves even though they don't smell the crab. So there's a bit of a theoretical paradox here where these cues are not thought to provide very much information about how prey should defend themselves, but on the other hand, they might be the only thing that the non-native snails can detect. And thinking about it from this perspective, could this information framework help us understand why recognition succeeds in some novel predator-prey interactions and fails in others? And if so, could it actually provide us a tool to help predict the probability that an introduced species could become invasive, or that an invasive species will have a different level of impact in different scenarios? So I conducted experiments to explore which information is important in risk assessment for our non-native noobs, asking if they are naive to a threat from native predators or if they're nervous, if they're twitchy in a way that ultimately could enable them to survive. And I totally got this working. Turns out you need the USB stick. And because I'm not totally crazy, from here on out, I'm talking about a subset of 4 of the 6 total invasive species. I think I might maybe stress Jen out there a little bit when she saw all those, did all this research she didn't know about. So sneaky. I looked at risk assessment in these 4 species, but I used only a single species of predator, and that's our native red rock crab. I put this picture up here not so much for diagnostics as to show you that prey might actually be motivated to hide when they detect this fearsome predator. And to zoom in on the salient feature, they have extremely strong crushing claws that can exert upwards of 180 pounds per square inch, which, if you are a small snail, feels a little bit like being sat on by an adult male elephant seal. Which is to say that these are capable predators of snails. There's a lot of evidence that they do control snail abundance and distribution in the Pacific Northwest. And so they are my predator of the day. So the general setup for this project was to test the defensive response of 4 species of non-native snail. And all of my non-natives are now going to be indicated in red. And I compared that to the behavior of 3 species of common native snail. All of the native snails are outlined in blue. This is a really nice group of snails to make this comparison. First, the group of non-native snails all share a similar length of history in the Pacific Northwest. They've all been here for about 100 years, even though they come from different regions of the world. However, the non-native snails are not known to be invasive elsewhere, although we also don't know if they've been introduced elsewhere. Second, both groups are phylogenetically diverse and represent multiple functional groups. So we have several predators here, and we also have grazers and deposit feeders. So for each of these 7 species, I ran a separate experiment evaluating their defensive behavior when they were subjected to one of these 6 Q scenarios. And I had the hypothesis that for native snails, more information should lead to a greater investment in defense. But for non-native snails, there could be this constraint on recognition of a novel native predator. So I generated a number of specific predictions about how these groups might vary. And I'll walk through those with you now. So first, I predicted that non-native snails would be naive to the crab predator. That is, the non-native snails wouldn't know they were in danger. They wouldn't change their behavior when they smelled chemical cues from that crab. But the native snails should change their behavior because they not only recognize it, but and in addition, it's a good indicator of risk. So this is the most basic assumption of naivete. Second, generalism. And this goes back to the idea that other researchers have suggested that these general cues could be advantageous for snails to respond to if they don't recognize the predator. So I predicted that non-native snails might respond to these alarm cues coming from conspecifics, but that native snails have access to better information, so they wouldn't respond as strongly. I had 2 more predictions about generalism that I wanted to use to sort of dig into that a little bit more critically. First, priority, that I predicted that because non-native snails only have access to information from crushed conspecifics, that they would prioritize those cues, they would respond more strongly than they would respond to information coming from the crab by itself. And the inverse should be true for native snails. They should respond more strongly to the crab than they should to alarm cues from conspecifics. Now, for all of the snails, the information from crabs attacking, consuming, and digesting conspecifics should be the most informative, the scariest information that they have. But for non-native snails, they might only be able to, out of all of those smells, they might only be able to recognize crushed conspecifics. So that alone might generate the same change in behavior as the full predation cue for non-native snails, but not for native snails. And then lastly, I had 2 predictions about those sort of the role of digestion in snails. And for me, it was less intuitive to figure out a priori whether there was going to be a difference between natives and non-natives, what the role of evolution would be. However, in general, I believe that digestion of the conspecific snails would increase risk assessment compared to if the 2 cues were just added together. So more chemical digestion information should increase risk assessment. And then secondly, prey identity is important to risk assessment. Namely, if you smell a crab eating one of your own species, that's scarier than if you smell a crab eating something else, which, as I mentioned in my case, was frozen fish fillet. That's right. Okay. So these experiments, each of these 7 experiments was conducted in polypropylene mesocosms, which is to say Tupperware shoeboxes. And in these shoeboxes, I observed the behavior of 1 or more focal snails, and I gave them a choice of what food to eat and if they wanted to hide or if they just wanted to cruise around. Those cue scenarios I manipulated upstream of the snails on which I took behavioral observations, so there was no actual interaction. In an upstream bin, I manipulated the cues by either including or excluding a single red rock crab. I could feed that crab crushed snails, or I could feed it frozen fish, or I could starve the crab, or I can add those 2 cues together. I can take the crushed snails and actually stick them downstream of the crab, so you're adding the chiromones to the alarm cues without allowing the crab to eat the snails. And I replicated each of those cue scenarios 8 times and measured how much the snails invested in behavior by recording the snails' location several times each week for over 2 to 4 weeks. At the end of which I tallied how much time the snails spent in avoidance. So were they trying to crawl out of the bin? Were they hiding under the refuge? Or were they just cruising around eating? These experiments were all conducted at Shannon Point Marine Center, where I, along with my father, where it was given free run for 3 years of half of their boatyard. It was an incredibly generous offer on their part, and I was really lucky to have it. Where we constructed these flow-through mesocosm systems, so you can see that the seawater, fresh seawater, is delivered individually to each of these bins. So having fresh seawater is critical to these experiments. It turns out these experiments on these snails don't work very well in closed or recirculating water. You get a little bit of a hyperaccumulation of the chemical information, which makes it harder for the snails to sort of discern what they should be listening to, get a signal among the sort of loud static. So from all of those shoeboxes, now you can witness the data. So I'm going to show you a series of graphs, one for each of the species, so each of the experiments separately. Then at the end, I'm going to come back and summarize in another graph the difference between natives and non-natives. So I'm not going to put any stats up here because that's not how I analyze the data, but I just want you to focus on the pattern for each species separately. So each of these bars is going to be the proportion of times a given snail was observed to be avoiding, and all of these Q treatments are arrayed in the same order of increasing information content, hypothesized information content. And remember that I would predict that for native snails, we would see an increase in the investment in defense when there is more chemical information indicating risk available. We don't know what the shape of that response is. It could be a threshold response where there's some minimum amount of information that the snails need before they change their behavior at all. But regardless, it should only increase, it shouldn't go down. So what do we see? So our first up is our native Nucella. Nucella was really helpful and compliant and gave me this lovely increase pattern in just the way I predicted, where you have these individual Q components don't increase their risk avoidance, so they're not spending much more time hiding when they smell either just chirobones or just alarm cues separately. But when you have both cues together, they really amp up their response. And for this species, it actually doesn't matter whether or not the crab is eating the snails, which is interesting. So let's see if this pattern was conserved, if the other snails were as cooperative as Nucella. This is the dove snail, Alia carinata. Alia is it's got a honey badger thing going on. It's really not intimidated by very much. It does increase its hiding slightly in response to unfed crabs, but really changes its behavior when it smells crabs attacking, consuming, and digesting conspecific. It's a huge increase in the amount of time they spend avoiding. Now, the last species is a bit of an oddball, so I'm going to spend just a little more time on it. Littorines, everyone's favorite hyperabundant snail. So littorines do this very strange thing, and I've never seen any other snail do it, where they decrease their predator avoidance behavior when they smell alarm cues from injured conspecifics. And it turns out this is true whether or not there is a crab present. So that's surprising, and it's confusing, and I'm doing other experiments to figure out what's going on there. You can ask me about it later. Suffice it to say, it is a repeatable phenomenon, and the thing I actually want you to focus on is that when the only time that littorine actually increases its hiding behavior relative to the control treatment is when crabs are allowed to attack, consume, and digest conspecifics. So there are definitely differences between the species here, which is what we would expect. But the salient sort of common thread here, I think, is the fact that they all require multiple sources of information before they increase their avoidance behavior, which is in line with the prediction that I was making earlier. More information leads to more certainty. So let's see what the non-natives do and if they give us anything interpretable at all. So our first is Atlantic oyster drill, some of which are out on the table if you're interested in what they look like. Closer up. So this species also really compliant. Isn't it funny how I put the biggest graphs first? So what we see here is, just ignoring this bar for a second, we see a really nice increase with information, but this glaring difference is that they respond very strongly to crushed conspecifics. They increase their behavior as much, their avoidance behavior, as much as they do to the full Q treatment. Those two are indistinguishable. So this is a really nice example of one way that we could have general cues being relatively more important for non-native risk assessment. Let's see if the other non-natives were as helpful as that one. So this is Batellaria, the mud snail, and actually, yes, again, we see that they increase their avoidance in response to alarm cues, whereas chiromones from the crabs are relatively less helpful, and this response to alarm cues is indistinguishable to when both cues are present. Godrilla, Japanese oyster drill here, also this U-shaped pattern where this snail spends a lot of time hiding in general, but there's still enough resolution to see that cues from injured conspecifics are just basically driving the entire response, the maximum response that we see. And 4 for 4, the eastern dog whelk also has a very nice U-shaped pattern where they respond very strongly to injured conspecifics, even though theoretically there's not that much information there indicating risk. So this is a really interesting pattern. These snails are from different geographic places, different phylogenetic backgrounds, and yet they all have the same, from my perspective, unexpected risk assessment pattern, where they're using cues that aren't very informative to determine when to protect themselves. And none of the native snails are doing that same thing. But I haven't actually showed you any analysis, so maybe you don't believe me, and that's understandable. I wouldn't believe me either. So I'll walk you through what I did to test the predictions that I talked about earlier. So I created generalized linear mixed models of the avoidance for each snail species separately. I used 2 implementations of that model to test the predictions. First, a 2-factor model that enabled me to test the separate and combined effects of the 2 Q components. And then I used linear contrast to target the other questions that I couldn't get at any other way. From those implementations, I extracted estimates of the model coefficients and variance and used those to calculate effect size for each comparison. And then the effect size for each species, for each prediction, I compared natives to non-natives with the t-test. So this will make a little bit more sense, maybe, when I start to build the figure that tests all these predictions. So to orient you to this figure, this is a plot where the parameter estimate from the statistical model is going to be shown by a single point for each species. I'm not going to leave this legend up here because I want you to focus on the difference between natives and non-natives, but I will pull out any salient species, any outliers that sort of bear mentioning. So anytime there's a positive parameter estimate, that means increased avoidance or greater avoidance on the positive side, and the inverse is true anytime there is a negative parameter estimate. And if the estimate overlaps 0, that means that there's essentially no effect of the comparison or difference between the 2 things being compared. So the first prediction was naivete, that only non-native snails would be naive to the crabs. And what this would look like is that the non-native snails would not respond differently between the crab and the control treatment, but the native snails should increase their hiding when they smell crabs. And I'm going to put some fake data up here just to orient you. So a positive would be increasing avoidance in response to the crab. And so I would expect, if this prediction were supported, that the native points in blue would scatter above the x-axis, so they might have different degrees of avoidance in response to the crab. But the non-native points should cluster around 0 because the non-native snails shouldn't be able to recognize crab. Predicted. So what do we actually see? We actually see that most of the snails cluster around 0. And there's no statistical difference between the parameter estimates for natives and non-natives. The striking thing to me here is this one non-native snail that actually had the greatest increase in its avoidance behavior in response to a theoretically novel native predator. And that's Urocylpinx, the Atlantic oyster drill, so the big compliant non-native snail I showed. Okay, so the next prediction was about generalism, that non-native snails rely on general cues, so they increase their hiding when they smell alarm cues from crushed conspecifics, but native snails don't. And so that would look like this, where the parameter estimates are positive for the non-native snails, but cluster around 0 for the native snails, indicating no effect of those cues. You might have some that have a little bit of a small response. There might be some situations in which it is advantageous to change your behavior a little bit. And this is pretty close to what we see. There is a significant difference between non-natives and natives, where non-native snails spend significantly more time avoiding when they smell alarm cues, and native snails don't. Cool. So priority. Do non-native snails prioritize alarm cues over crab cues? And the inverse being true for native snails. So if it's positive here, they have a stronger response to crabs than crushed conspecifics, and if it's negative, they have a stronger response to crushed conspecifics. So this is the prediction that non-native snails would cluster closer to conspecific cues and native snails would cluster closer to crabs. And we have something that's pretty close to that. So first of all, the native snails significantly favored crabs over crushed conspecifics, and most of them, well, they either increased their response to crabs, favored crabs more strongly, or they didn't have a difference between the two. This species, incidentally, is Nucella. However, all of the non-native snails responded more strongly to crushed conspecifics than to crabs. And now sufficiency. Again, this prediction had to do with non-native snails. The only thing that they can even smell is crushed conspecifics, so that response should be the biggest response they ever give us. And if that were supported, it would look like non-native snails can't tell the difference between the full predation cue, including snail-flavored crab poop, and the crushed conspecifics by themselves. But native snails have varying, again, some sort of spread here, but they generally will increase their avoidance more when they smell more chemical information. Here, we also have a lovely difference. It's almost like I hypothesized it perfectly. So the native snails have a significantly larger parameter estimate than the non-native snails, which all cluster around 0. So now the last two predictions were related to digestion, and remembering that I didn't have a specific prediction about the effect of coevolution here, but that both native and non-native snails were expected to spend more time hiding when they smelled crabs attacking, consuming, and digesting snails of their own species than when just the alarm cues and the crab cues were mixed together. And what that would look like is this. You'd have overlap between the species, no real distinction between natives and non-natives, but that they'd all favor the full predation cue. This is not exactly what we get. In fact, almost all of the snails clustered around 0, meaning they did not require digestion to demonstrate the maximum response, which was a little bit surprising. But there are two notable exceptions, and I'll put those up here. So our weirdo Littorina, which actually, as I mentioned, reduces hiding compared to the control whenever it smells crushed conspecifics, unless there is digestion. So for this species, digestion completely reverses their change in behavior. And then honey badger here, which does actually increase hiding a little bit in response to unfed crabs, but really needs information from digestion in order to know it's in danger. So this, even though those two species were different, this was not enough to sort of swing a statistical difference between natives and non-natives in the parameter estimate. And then lastly, the effect of prey identity, that if you smell a crab digesting one of your own species, that should be scarier than if you smell a crab eating something else. Again, I hypothesized that both natives and non-natives would be more scared when they smelled crabs digesting one of their friends. And in general, that is what I found, that natives and non-natives were on average similar to each other, but also that they were positive, meaning they did increase their hiding in response to snail-flavored crab poop. And Alia is this species which was really scared of snail-flavored crab poop. But in general here, no difference between natives and non-natives. So to summarize that sort of huge plot I just put up there, I didn't observe the support I expected for naivete, mostly because very few of the snails actually demonstrated a response to crabs at all. And the one snail that demonstrated the strongest response was non-native, so that's a little bit strange. However, I did observe support for generalism, and non-native snails not only did respond and were distinct from native snails in defending when they smelled alarm cues, but they also responded more strongly to those cues than hypothetically more informative cues from a crab, and non-native snails did not. And those cues were sufficient to elicit the largest response that I observed out of any of the treatments for every species. Now, in terms of digestion, Digestion was actually not required for the majority of the species. It didn't depend on whether or not the species was native. However, it was very important for 2 out of the 7 species, both of which were native. And then lastly, prey identity, having a conspecific prey digested, always increased defensive behavior for snails, but that didn't differ between natives and non-natives. So to draw out the salient point here, or what I think is the salient point, I do find that these predictions associated with information generalism are all supported. Non-native snails were the only ones to demonstrate these traits, and they all did, and native snails never did. So this set of experiments is the strongest evidence that we have to date, or at least that I'm aware of, that test this hypothesis that non-native snails could circumvent naivete by using different information that they already have evolved to recognize. It also refines the definition of what an information generalist might be. So generalism is thought to be pretty important in biological invasions, or it can be a trait that favors success, because if you're not picky about what you eat, you can show up and basically find something digestible and some tolerable couch to crash on. But it's possible that this information generalism could also favor prey that show up and don't recognize a predator. So this could play a role in invasion dynamics. However, one of the outstanding questions that needs to be answered before we get too carried away with these things is whether or not the generalism is a driver of invasion, or it's actually a response to the introduction event. So generalism could be a trait that's present in the population that predisposes them to be very good invaders before they even get here. But it also could be selected for by the introduction event, essentially a bottleneck on the initial propagule. And I've tried to do experiments that get at this. This was a set of experiments I did where I actually imported snails from their native range. Well, it's not unfortunate, it's completely appropriate that when you do that, because of concerns about shellfish disease, you have to do those experiments in quarantine. So I can't do them in the flow through seawater, and these snails, for these species of snail, that makes it just essentially intractable. So I think the next step in this case would actually be to test the snails in their native habitat on the East Coast, or maybe in Japan. I'd have to do some preparation before going there. Nevertheless, the other point that I want to make is that there are situations in which being overreactive can be favored for the snail, can help them actually overcome a problem. However, I put as a caveat, at least temporarily. And the reason is snails might be surviving here because they have this information generalist trait. But they could also still be incurring a fitness cost relative to snails that require more information. So this actually has a name. It's called level 4 naivete. So level 1 naivete is that, according to these guys, is that they don't recognize the predator at all. Level 2 and level 3 have to do with they recognize the predator, but they either have an inappropriate or insufficient defense. And then level 4 is that they recognize and respond appropriately, but they're actually responding too strongly, in which case they're still incurring a fitness cost. So all of these are suboptimal fitness scenarios. So if that's the case, and non-natives can evolve the ability to recognize crabs, which is not necessarily a given, information generalism should be something that's selected against over time. So this is something that we might see playing out over time. There are also a few tests that would be an even stronger test of this information generalism theory. So I compared successful responses of successful invaders to responses of snails that are not known to be invasive elsewhere. An even better comparison would be to compare successful invaders to failed invaders. It's notoriously difficult to capture invasions that fail all on their own. So it'd be important to select a system in which you could identify individuals that have failed, or species that have failed. The other question is whether generalism is actually itself generalizable. And I have Hillary to thank for this from years ago. Yeah, she's quizzical. Hillary had this idea, but yeah, I'm blaming all of this on you, evidently. Sorry. Now, it's a great idea. So these snails respond to alarm cues from crushed conspecifics individuals of their own species, but is it possible they could also recognize alarm cues from other species? Does that ability to use alarm cues from other species, is it stronger in non-native species than native species? Is it constrained phylogenetically? Is it constrained based on evolutionary sympatry? These are all sort of big, meaty questions that would make a really good dissertation for someone. And then third, does this matter for communities? Does it change ecological dynamics that one species requires more information than another? Animal personality is increasingly getting recognized as a driver of ecological dynamics, and is this one place that that could also play out? Okay, so I think the other salient point that deserves at least one slide is this idea that very few of the snails actually responded to the crab itself, when I hypothesized that that should be pretty decent information. Of course, there was the glaring exception of the Atlantic oyster drill, which did and showed the strongest response to the crab. So this is a pretty smart snail, and I'm curious about how it got so smart. I think there are several potential explanations. So rapid adaptation of the population following introduction, an exaptive ability to recognize the crab before it got here. Possibly they show up and they already know that something called crab is scary. And associative learning within the lifetime of an individual could also enable recognition. It turns out that my entire dissertation was supposed to be focused on disentangling these three mechanisms. But as I mentioned, some of the experiments turned out to be intractable. And so this remains an open question if we can find the right system to answer it in. However, even though it's interesting that this snail does recognize crabs, it's also surprising that most of the snails, even the native ones, did not increase their defensive behavior in response to crabs. It's not because they're naive. We know this species responds to Cancer productus. It thickens its shell. So why wasn't I able to capture a defensive response? Well, I was intentionally oversimplistic when I presented this sort of information specificity axis. So it is important that prey know where a predator is and what kind of attack they might expect, but it's also important that they know whether or not that information is actually associated with risk. So the prey might be getting very, very specific, a lot of information, but that information might tell them that it's totally cool and they can just relax. Alternatively, they might be coming across a cue that is always associated with getting attacked by a predator. So it might be a high indicator of risk, but it might not tell them how to respond, in which case, what do they do? So we could theoretically array my cue signals in bivariate space and make more nuanced predictions about what to expect, with the general expectation that the probability or investment in defense should increase as you have more information that indicates greater risk. Certainly at the upper end of the scale, if you're certain you're about to be eaten and you know what to do about it, you should do something. But I think one of the limitations or the challenges in this framework is figuring out how to ordinate the cues in terms of risk. So how do you determine how much risk is actually reflected by a given information signal? I think there are some first blush approximations we could make. For instance, a fed predator is probably less dangerous than one that's hungry. And so, I'd rather hang out with a tiger after dinner than before. And maybe if a predator is eating something else, it specializes on something else, so it doesn't pose as much of a risk to you. But I really don't know what you would do with alarm cues in this. It probably is very system specific. That is, if you smell a crushed snail of your species, is it always because that snail has been crushed by a crab that's going to come for you? Or is it possible that snail has been crushed by a rock being moved by a wave or a boot on the beach? So I think it's a really difficult challenge to figure out where you would put these cues a priori. I think it's also a really interesting experimental challenge to figure out how you would tell which cues were actually associated with risk. And complicating this even further is the idea that the probability of responding is constrained by ecological, evolutionary, and environmental contexts. For instance, cues can have multiple selective forces acting on them in opposite directions. This is a barnacle that has a cuticular protein that functions not only as a settlement cue for larval barnacles, but also as an attractant for predators. Prey motivation state also says something about whether or not they will respond. So if you're hanging out by the river, you might prefer to wait until the crocodiles move off before you go in, but you might be so thirsty or so in need of migration that you have to take a risk you wouldn't otherwise want to. And the community of other prey affects your need to invest in a defense. If you are in a school, you only need to outswim the other members of your school in order to survive. So your response might be stronger if you're on your own than if you're in a group. And evolutionary novelty is also one of these constraints. So this is all to say that even if we could figure out where those cues fit in this bivariate space, the landscape is actually probably much more rugged than that, where you have responses that are constrained by multiple trade-offs, even though you're not given very much information. And you might have constraints against responding because you don't have access to information, or again, there's some other selective action on that information. So I leave this out there as a theoretical challenge to be addressed by future generations. I feel like that this has really only scratched the surface of what evolutionary novelty can teach us. Biological invasions have a lot to teach us in terms of the role that evolution can play in species interactions. And some of the really interesting, outstanding questions are, how long does novelty last? I've had a lot of people ask me, when does an invasive species become a native species? And I don't think there's really an answer to that, to be honest. And how novel is novel? Is it a difference in degree or a difference in kind? Both of those could be true in different scenarios. And then, does novelty affect all interactors in a situation equally? Or is it better to be an invasive prey that's facing a novel native predator? Or is it better to be a native prey that's facing an invasive prey that's facing a novel native predator? You know what I'm saying? You switch out? Okay. I need to draw a diagram. So in any case, so these are some of the grand challenges that I see to evolutionary novelty and what it has that it can potentially teach us. So I will leave you with these questions, and I hope you'll indulge me for just a few more minutes before your hands shoot up with questions, which I'm sure you're all brimming with them. And allow me to thank the additional folks who have made this work possible. So first, I want to take a second to thank the folks who have put their pocketbooks and wallets behind this project. So I received generous support from the NOAA National Estuarine Research Reserve System, GRF, and was really a part of the Padilla Bay NER community, which was great. Doug and Suzanne in particular helped me a ton, not only with doing my work, but it's just so nice to have people who are interested in your work, and they were always super great. I mentioned Shannon Point as being critical to my ability to do those mesocosm experiments. They were, again, extremely generous logistically in helping me plan and just tolerating my general being there and making a mess on an ongoing basis. All of the folks who supported the construction of the mesocosm are actually memorialized up there at Shannon Point, so they can go and see their plaques. And in particular, I'd like to draw attention to the Pacific Northwest Shell Club. They've provided multiple grants in aid of my research, which has been fantastic. But I also am extremely appreciative of how warm and welcoming they've been into their community. They're all extremely knowledgeable, engaged, passionate people, and it's been a very warm community. These are the people who discover most new species of mollusks in the state of Washington because they are out there and they know exactly what they're looking at. So I have really been lucky to spend time with them. I think some of them are listening, so thank you. I wanted to thank the Department of Biology for also providing a number of awards, as well as Puget Sound Restoration Fund and the National Shell Fisheries Association. I need to thank all of the people who provided hands and muscle and sweat equity into my work. All of the people who came on field adventures with me, and who helped me count snails, find snails, crush snails, feed crabs, etc. I have to thank the folks who also helped me with a lot of the logistics. Doing marine research in the field, you rely on the good faith of a lot of people who live on the shoreline to give you access. And more often than not, I met some really lovely people. The Gitches had me in for coffee every time I was out in their neighborhood, which was really nice. And Danny at Taylor took me out to the oyster beds in the middle of Samish Bay multiple times in the middle of the night in questionable conditions, which was like, I wanted to go. Like I would have been mad if he wouldn't let me go. So I appreciate him taking that personal risk and potentially liability risk of taking me out there with him. It always worked out fine. And Brady at DFW for helping me get through the importation process, and Puget Sound Restoration Fund for giving me access to the restoration beds. And the RUSync lab and the extended lab family, I love our lab family because it is so inclusive of all of the marine folks, have just been excellent at providing pastries and ice cream from time to time when it's been appropriate, as well as a ton of intellectual support and moral support and just enthusiasm for the topic overall. I left this big blank space here to draw special attention to the mesocosm master technician, my dad, who not only helped me build this large piece of equipment and embarked on the adventures of learning how to plumb systems somewhat effectively, but also stuck with me through scrubbing and washing 210 bins 7 or 8 times, and all of the pipes and fittings, because all of that stuff has to be washed in between experiments. So he spent many hours under my thumb, allowing me to practice my tyrannical leadership style, which I am really appreciative of. So since he's not now volunteering for me, he's available for you guys if you need. I'm just kidding. He's volunteering for my other projects. He's taken. So also there's a lot of hearts and brains behind this research as well. My one brain and my one heart is really just not enough. First, I want to thank my collaborators on some of these, Sean and Eric, who are both kind enough to be here today. Thank you both. I'm going to move so I can see Eric too. Thank you both for your ideas, for your data, for your enthusiasm, for your equipment, and for keeping the faith through multiple rounds of really difficult revisions and resubmissions. Positive attitude is a huge asset on a team with me. I want to also take a moment to sort of thank the legacy that we get from Bob Payne. The intellectual legacy, particularly in our lab, has been really valuable. My work, reeks of Bob Payne in a good way. And in particular, so Bob is, of course, a global influence, but he is also directly involved in students' work. And I've had a lot of great conversations with him. He was involved in our fall ecology seminar, where I think it was like the ecology of fear, something like that, which was helped me put together a lot of the theoretical framework and chew through some of the ideas that I presented here today. I have a ton of people who have contributed to maintaining my sanity over these last 6 years. Your cohort is the most important group of people in your graduate experience. And I always tell people when they're applying to graduate schools that you should really be interviewing the people who you're interviewing with, because those are the people who are going to get you through. So thank you very much to my cohort. We are the best cohort, just saying. But in particular, I also want to draw out that a number of graduate student ladies who are just awesome women and who have saved my bacon on a number of times in a number of ways. So thank you guys all. And also the other folks who have been instrumental in keeping my sense of humor and enthusiasm. So it's easy to get burned out on snails, turns out. I don't know. Actually, Hillary might beg to differ. I'm sorry. I don't. But it's easy to get burned out sometimes, and just in general as a graduate student. And so the people who keep your enthusiasm about science going are just incredibly valuable. And the people who've shared an office with me, thank you for tolerating my whatever. So I'd also like to thank my committee. I added an honorary committee member who's not on the books, but Ben Miner was my master's advisor and has continued to be invaluable to my research. He's not only volunteered his students and his friendly reviews a number of times and just his brain on analyses, but he's also just always said exactly the right thing at exactly the right time to me, and it's just been it's not quantifiable how valuable that is. My on the books committee, Julian, who is my GSR, has been an exemplary role model in terms of scientific rigor, productivity, all done at an incredible pace and with a positive attitude. So I am grateful to have him in my court. I think you're in my court, right, Julian? We'll see. And Emily, thank you very much. Your spirit of adventurism, that's not a word, you know what I'm saying, spirit of adventure, and also positive attitude, and also just helping me to feel confident in my own science has been a huge asset. I'm so glad you made the trip. Emily came from DC this week, so I'm very grateful for that. And Ben, you also, I also,
Positive attitude.
I know, I was like, oh, okay. So Ben also has a positive attitude, which is hugely positive. Everyone, I think, in this room knows what a great human Ben is, with so much integrity. And I think the thing to me that is the most important is the respect that you treat me as a graduate student with, and my work, the value that you put into it. So it makes me feel very good. Thank you. And my advisor, Jen. Jen, thank you for being down with me in the mud on my knees in the rain, looking for snails of exactly the right size. And then after that, spending 6 hours tying them to pieces of rebar. Thank you for that. Thank you also for helping me clarify my thinking. Anytime I don't know what it is I'm thinking, I'll go to Jen and say some words, and she has this incredible power to distill something that makes sense from everything that see, Alex is laughing because he knows this. This is like this. From everything I just said, she distills reason and logic from it. And she's been an incredible role model also in that, helping me know it's okay to balance my life. And I think I would not have made it through graduate school without that. So thank you. And of course, the folks who share an everyday life with me, most of them, not actually most of these, some of these are too far to share an everyday life with me. So my family, thank you so much for raising me with a love of knowledge, a value of hard work and pursuit of knowledge, a sense of dry humor that appeals to some people, and certainly a sense of humility. Matthew, looking at you. And also to my local partners, partners in crime, who make me coffee and take care of me every single day, and have taught me unflagging compassion for my struggle, and have been able to share excitement in science, but also provide me distance from it. It's helped keep me on the track to actually finishing in fewer than years. So thank you guys all for being here. Thank you for indulging me, and I'd love to take any questions if you have them. Thanks. And I know I kept you a little bit long, so if you do need to sneak out, no shame. Yeah, Brandon.
First off, awesome. The snail that comes out of hiding when it smells kind of specific. Do you know any natural history? Is it a cannibal or anything like that?
So,
is that a food opportunity?
Right. So we've tried to test cannibalism. We need to sort of do this again. So just actually looking at whether they're coming out of hiding because they're attracted to the smell. So are they moving towards injured conspecifics? We don't find any support for that. I had this other idea that maybe they can't tell those other snails are dead or injured on their way to dying. And so maybe they see it as a competitive threat. I should get out and about and eat before everything gets eaten. But it turns out they can tell what the difference between crushed and non-crushed snails, and it's density dependent. If you have more crushed snails, they come out of hiding more. But if those snails are live, they don't really change their behavior at all. So I still don't know. Eric's theory is Purple Heart. He calls it the Purple Heart theory, that they're going into the fire to sacrifice themselves for their friends. I haven't. What's that? Sorry, Eric. I didn't mean. Yeah. No takebacks.
That's a nature paper,
yeah. Yeah. Right. I think that would be a nature paper. So yeah, if anyone here has theories that we can test, I think we do have some more tests that we want to do. Is their defense to just move around, increase their movement randomly in water? So we're going to do some video studies to see how they're actually take a longer term look at how they're using space. If there's other ideas, I'm happy to entertain them. Jake?
I love the Christian generalist idea. I know it's not tractable to take species from Japan, say, who did not invade and test them, but could you take conspecifics of invaders from Japan who did not go through this bottleneck and 100 years of selection, especially that initial selection for ability to invade?
Yeah, so that's actually what I did, or that's what I tried to do. So there's some snails out on the table there. Those are snail shells that are actually from Japan. I had them import the same species, and I compared it to our local ones. Is this what you're? Okay. These are the species that you have to keep in quarantine. So I did these experiments essentially I tried them like 4 times, but either in large 150 mil centrifuge tubes, and you can look at sort of their vertical migration, or I did them in small bins and looking at hiding versus no hiding. But I think they just don't work in closed systems. Yeah, so they could be carrying some shellfish disease that could be transmissible to our local snails. So it's like serious quarantine with bleach and everything. So yeah, you're on the right track. I want to do that too.
I thought the quarantine was just for snails that had not already.
See, that is also true. But yeah. Yeah, Jason.
So, really nice, thanks. I was wondering about the fact that for both the non-native and the native snails, there was a lot of variation in terms of how in your control, how much time they spent out hiding. And I was wondering if like, you just look at that as a feature. Is there anything? I mean, are the ones that basically have less of a scope, as you described, for being able to go out towards and see what they decide to do?
Yeah, I think that's a good question. I've never actually just taken the control treatment and say, okay, well, how much time do they spend hiding? Is that sort of what you're suggesting? And whether it differs based on, whatever, fill in the blank. I want to sit down and think about that. Do you have any sort of thoughts? I mean, there are snails, I think, like the predatory snails have a pulse feeding strategy. So they go and they get a barnacle or a whole oyster at once, and maybe they don't need to eat for a while. So their baseline level of hiding can just be higher. And then I think this is true. I'm not actually sure. Somebody in the room who knows more about snails, I'm looking at Emily and Hillary, can correct me on the need, how frequently other types of feeding strategies need to move out and about. I think that's one relevant comparison. I'm not at the moment able to come up with anything that might suggest a difference between natives and non-natives in level of hiding a priori as a hypothesis, but I'd be curious to see what actually happens. Thanks. Yeah, Kevin.
That was a great thought.
Thanks.
My question is whether you have any idea for the non-native snails that don't seem to cue in on that crab signal. Any sense on whether they would cue in on a crab signal from their native region?
Also a good question. So, I don't know. There's no evidence. There's no research out there. There are crabs of the genus Cancer in both of these native regions. So there's some debate about whether or not, I know more about the Atlantic oyster drills, whether or not Atlantic oyster drills experience much predation pressure from Cancer crabs, because Cancer is more subtidal over there, I guess, and Cancer is also sort of subtidal here, although Productus is somewhat upper subtidal, lower intertidal. And so I don't, there's no evidence yet. I think it's an obvious next test. That's what I'd like to do with, I don't, not today. But I think it's an obvious next test for, this sort of generalism. Chris. Oh, nice. Good teamwork. Yeah, Beth.
Given you spent a lot of time on your dissertation in the field, I'm wondering if any of your requests for individuals in the field, any of your observations, I guess, complement some of your findings in terms of where you found them, or potentially in areas without more.
Yeah, so the first thing that comes to mind, is actually Atlantic oyster drills. So the area in Willapa Bay where Atlantic oyster drills are most populous is actually, and probably Eric and Jen could speak to this, is the place that I go to collect them because they're the most abundant is the south end of Long Island, which is relatively fresh. So Cancer productus is less abundant there than I would expect it to be elsewhere. That's just the very broad brushstroke. Most of these species were collected from beaches that actually had Cancer productus at them. I could collect those crabs at the same place. I'm less certain about the other. I mean, it's interesting that is the snail that has the strongest response to crabs, but that's as far as I think I can get with that at the moment. I'll think about it some more. Thanks. Anyone else?
All right, well, let's thank Emily one more time.